Okay, so we are looking at the cell cycle. A lot of information kind of crammed into one, but you guys will have a good chance to practice a lot of these and really see the process come together with what we'll be doing later on this week and through next week. Okay. So let's go ahead and start by looking at why do cells divide in the first place. We know that the DNA, that genome, has all the information that is needed to build a cell, to build either the proteins or the different RNA or whatever functional thing there is coded for by the DNA, which means that we need to make sure that the cell is going to divide and have that information present. So why even divide in the first place? Well, if you are a unicellular organism, a couple of reasons for that. And they kind of go hand in hand. So usually what's going to happen with unicellular organisms is that their surface area to volume ratio decreases as they grow. So you'll remember as we were looking at cell membranes, that membrane is going to be how stuff gets in and out. So as a cell gets larger and larger and larger, the volume grows. So that volume is going to get exponentially larger compared to the membrane. Okay, so as these cells grow bigger and bigger and bigger, they get less and less efficient, and they finally reach the point at which they have to divide in order to continue to function. And that is also for unicellular organisms, that is their mode of reproduction. You start with one, you end up with two, reproduction, okay? In multi-celled organisms, we're gonna see the same thing. If a cell gets too big, it's gonna have to divide for the exact same reasons. It's not a very efficient thing to be too big, but not have enough membrane to allow materials to move in and out and get the materials that you need. Some multi-celled organisms are also going to use cell division for reproduction, but it's not going to be all of the cells in the multi-celled organism. So for example, in humans, we don't reproduce with skin cells. Obviously, please tell me you knew that your skin cells do not make babies. So it's only going to be a select few that are capable of that part. But in multi-celled organisms, the process of cell division is going to have a whole bunch of new purposes that aren't the case in unicellular organisms, such as growth, right? I know that me and a mouse have the same size cells. The only difference between us is how many we have. And you know, just the general genes that a mouse have would be way different than mine, I hope. I don't know, whatever. As organisms are growing, they're also going to have their cells differentiating, meaning going from a very unspecialized, not very capable, but can be anything when it grows up kind of cell, to one that is very specific, very fine-tuned to do a particular task. So each of the different tissues in a multi-celled organism will have needed to be specialized or differentiated so that it can do the job of whatever its tissue or organ is. And then of course, repair and replacement of cells. You are constantly shedding your skin. Your poo is like a quarter dead skin cells from your digestive tract. Yummy, right? And also anytime you hurt yourself, you're going to need to do cell division in that area so that the tissues can be repaired, so that the cells can patch up that area and you can be good to go. Okay. So we'll start by looking at prokaryotic cell division because this is fast and easy. And the reason that it's fast and easy is because the prokaryotic structure is a whole heck of a lot easier, right? And so we're going to start by taking a look at this stuff in here. That is the chromosome of the prokaryotic cell. And we know that it is not encased in a nucleus. Instead, it is in the nucleoid region. All right, and so this is where we have to really look at the difference between prokaryotes and eukaryotes. This is where it really comes into place, is that prokaryotic cells are only going to have one chromosome. All of their genetic information, all of the genes that they need to make themselves and to be able to function are going to be found on that one chromosome. Okay, and so why does that matter? Hold on, let me add one piece here. So it's going to be all the genes that are necessary for it to survive. Do prokaryotic cells have any other genes that maybe aren't necessary, but like they're there? If you want to write that in the chat, if you can remember what those little pieces of extra DNA are called. Or if you just want to write IDK, so at least I know you're, still awake. All right, we'll help you out. There you go, nice job, Chris. So plasmids are going to have non-essential genes, okay? Meaning that if you're a bacteria and you're just chilling and you have some extra plasmids hanging around, great. A lot of times they may have stuff on them that are really useful for like antibiotic resistance, for example, or might help them adhere to one another better, but they can still function without them. And the reason that I bring this up is because if they do not have plasmids, it's not the end of the world for them unless they're in an antibiotic environment, but that's besides the point. All they really need are their chromosomes, that one single big old chromosome, and they can survive and they're good to go. Okay, which means for them to divide is a really, really simple process. If I have a piece of paper and I photocopy it, and then I need to take that and make two piles that have exactly the same information in it, it's really easy to just be like, all right, pile one, pile two, done. And that's really what's happening in this process that is called, I'm going all over the place here, binary fission. And so because of how simple these prokaryotes are, their cell division can be hecka simple as well. So it starts with what is called the origin. And that is a little part of the DNA of their chromosome that starts to separate so that new nucleotides can be added and the DNA can replicate. So we take that genetic information, photocopy it via DNA replication, and by the end of that process, the one single cell will now have two sets of genetic information. And that origin essentially is now integrated into both chromosomes, and it'll move towards the outer edge of the cell. And they'll continue to push outward until the cell grows really, really wide and can pinch off in the middle. Boom, we're done. So binary fission is going to be a process that happens really fast, as in if bacteria could grow at their maximum rate and not be limited by the nutrients, then they would basically be covering the earth 8 miles deep in a matter of a couple of days. So they can do this roughly every half hour, they can double. And if that continues without any limit, they can reproduce so quickly that they are completely covering the earth, even though they are only microscopic. And so a really efficient process when it comes to us thinking we're so cool because we have a nucleus. Oh, guess what? Even without a nucleus, these guys are really able to dominate because they can replicate so quickly, unless we give antibiotics and it ruins their replication. Moving on. Okay. Now we compare that to what eukaryotic cells have, which is way more complicated. And so let's go ahead and first look at what a eukaryotic chromosome looks like in terms of what the DNA is, and then talk about why we have some issues and can't really do that process of binary fission. So right in here, I've got DNA, and we can see that it kind of coils around these. I want you to think of these as little spools. If I had a whole bunch of thread that was just unwound, it becomes really difficult to separate it from everything else. So by winding it around these little spools here, they're called histone proteins, but we'll see them again later. I don't need you to memorize that now. And then we start folding all of those histones into a coiled mess going on here. And that coil supercoils until eventually we end up with chromosomes, okay? So we've got the DNA in its relaxed phase, and then it coils, and then it supercoils, until eventually it's really, really condensed. This part gets confusing. The term chromosome can mean a lot of different things and a lot of different forms. So when we say chromosome, what it really means is DNA and the associated proteins that come with it. So one big honking piece of DNA and all of those proteins. And so it can exist in a really loosey-goosey form. It can exist really, really condensed, and both of those are technically chromosomes. It can even exist double, and it's kind of technically a chromosome. So to really clarify what's what, we add a whole bunch of different terms to mean the chromosome in different states. But it all sounds the same, so it makes it kind of harder. Whoops. So in a non-dividing cell, in a cell that is in what is called interphase that we'll look at here in a second, the DNA is relaxed. And the reason is so that different enzymes can come in, land on the DNA, and start using it, start using that information for making proteins or making different RNA. And so the DNA in that really relaxed, loosey-goosey form is called chromatin. That's what I need. There we go. And that chromatin really spreads out throughout the entire nucleus, and each chromosome kind of takes some of its own space, but it's really, really loose and just chilling in there. When it is time to divide, that chromatin is going to condense. Going back to my thread or yarn analogy, imagine having 46 different spools of thread that are all unwound and just put into a barrel. Imagine trying to separate those out. It'd be really, really difficult. Actually, 92. Anyway, no one cares. Separating out even 2 things of string would be incredibly hard because they're going to wrap around each other. But if it's already nice and spooled up, all you have to do is be like 1 thing of thread, another thing of thread, boom, done. So that's what's going to happen is these DNA chromatin molecules are going to supercoil up and condense. Okay. I wish there was an easy way for you to give me a thumbs up without me turning on all the pictures. That's okay. Tell me how much sense that made, 1 to 5. 5 is like, okay, you're following where I'm at. A 1 is like, you are very confused. Just put that in the chat for me. To the people that cannot make a decision between like, I'm kind of like a 1.76. If you're using decimals, you're making me laugh right now. Okay. All right, so I'll make sure that as we're going through, I'm really careful about which term I use to mean this piece of genetic information, okay? All right, so let's talk about this really condensed DNA. This really, really condensed molecule of DNA and other proteins called a chromosome, when it is relaxed, when it is in this stage over here, is when DNA replication is going to happen. So we'll come back to this in a little bit, but I want you to like make the connection in the visual piece going on, okay? So here is the genetic information. When a cell is going to divide, it's going to double up all of that genetic information. So by the time it coils, there's already 2 copies of it, 2 photocopies of it, right? They're completely identical to each other. And that's why we see that in the condensed form, we usually have these 2 kind of stuck together. And so those are going to be called sister chromatids. So tid at the end there. Tin, think noodles, right? Chromatin is all loosey-goosey. The chromatid is not, I know, I fail. And so what we have here is really 1 molecule of genetic information, which has been perfectly copied so that now we have 2 pieces of genetic information that all have the exact same information on them. And so sister chromatids are going to be identical to one another, meaning all the same genes and all the same alleles. There's 1 chromatid, and there is the other, okay? And these are held together in the center by a centromere. All right, let me go back up to the previous slide so that now we can look at where the complication comes in, because we do not have 1 chromosome that then just gets doubled and then just has to get separated. We have 23 different chromosomes, and we have 2 sets of those. So these are those homologous chromosomes, right? A pair of homologous chromosomes. And remember, those are the same genes, but potentially different alleles. So what you got from your mom and what you got from your dad. For chromosome 1, that makes a homologous pair of chromosomes. So in a non-dividing cell, in a cell that is just chilling, you have 46 molecules that we're going to call chromosomes. Okay. The chromatin that is all loosey-goosey in your cell is 40. Did I say 26 a minute ago? I meant 46. I'll say that again. 46 different molecules that are all splayed out and just relaxing, that then get doubled up to make 92 molecules before they condense. Okay. So in the condensed form, we have two molecules that are kind of stuck together. Each one of them is called a chromatid, and they're held together. Sometimes you'll see each one called its own chromosome. Sometimes you'll see the pair of them together called a chromosome. Now you see where the confusion comes in. So we're just going to call that pair a pair of sister chromatids. All right, another 1 to 5. Tell me how much I'm making sense. 5 is like, okay, you're following. Oh, I'm seeing the first few numbers coming in are already less than where we were a minute ago. Oh man. 3.7, thank you. Like 3 point something. All right. Let's see if as we go through, we start to make a little bit more sense, okay? So let's talk about the eukaryotic cell cycle. The whole point of this is to take out all that replicated, that doubled stuff, and make sure that it is properly sorted. Think of it this way. I'm going to keep coming back to cards. I'm sorry. Fun fact, I was born in Vegas, so like, of course, I'm going back to playing cards. Duh. So if I was to take 2 decks of cards, let me rephrase that. If I was to take a deck of cards and replicate it, so now I have 2 identical decks of cards, and then I shuffled them in together, I'd have 104 different playing cards, right? Because each deck has 52. That's kind of like the 92 pieces of genetic information that you have in your cells before they divide. What we're going to be looking at is how are they then sorted out one by one. So having them as sister chromatids would be like going through and finding the ace of spades, putting them together so that later when you go to sort them, they can be paired out evenly. Okay, so let's look at the 2 parts of a cell's life cycle. Really 2 main parts. We have what is called interphase, and during this period of time, the cell is not actively dividing. And this is when cells are going to spend most of their time. Thank you, Alex. They're usually going to be in interphase, except for ones that are very rapidly dividing type cells. But some cells never really get out of interphase once they're born, right? So we start with an itty bitty baby cell. It helps if I have my marker and not my eraser selected. Duh. Okay, so we start with this itty bitty baby cell right here, which is going to grow and grow and grow and grow and grow over the course of interphase. And so by the time it is a big mature cell that is now losing efficiency, it is going to go through a different stage to actually divide. And that different stage is called the M phase. So interphase is when it is not actively dividing. M phase is when it is. And so at the start of M phase, we're going to have one big ol' honker cell who's ready and raring to split. And by the end of it, we have 2 new baby cells, and they're each ready to go on and interphase and live their lives. Okay. All right, so when we look at interphase, let's break that down just a little bit, because as it turns out, interphase is broken into 3 parts. And those 3 parts are going to be called G1, S, and G2. In a G1 cell, that's called the first gap, and all the cell is really doing is like growing and hanging out and doing its thing. It is not inactive. This is not like some period where the cell is just like, hey man, I'm chilling. It's still doing a ton of transcription and translation, protein synthesis, making stuff, right? It's still doing cell respiration if it's a plant, photosynthesis. It is really busy. It's just the one thing it's not doing is actively dividing. So we call this the gap 1, and we just have like normal stuff happening. Okay. A cell that goes into S phase is committed to continuing through the rest of the cell cycle. So once you get out of G1, once you move into the second phase of interphase, you're going to finish it or you're going to die trying, literally. Okay. So S is for synthesis, which is when the DNA is replicated. So it's still all loosey-goosey in the form of chromatin. All throughout interphase, it is chromatin. But during S phase, now I've got twice as many molecules of DNA. So let's go ahead and kind of take note of that, is that in humans, we're looking at 46 pieces of genetic information, 46 molecules, 46 chromosomes, but I hesitate to say that. And then by the end of S phase, you are at 92 pieces, chunks of DNA. Doesn't that sound weird? Chunks of DNA. Sounds like a weird 90s band. Anyway. And then we get into G2, that is the second gap phase. And G2, gap 2, is going to be when the cell is doing its final preparations to make sure that it is good to go for the other phase, for M phase, when it is actively dividing. And so some of the things that it's going to be doing are prep for M, doubling the organelles. Organelles, look, I can fit, yay. And just checking over everything to make sure that there are no issues in the cell. If there are issues, we got to go back a step, make sure that everything is good. And if it is not, then die. But if we're good, we can go on to M phase. Okay. So now let's look at the M phase, because this is where it gets confusing. M phase is really, really short, but it's really divided into two parts. Number one, let's get that nucleus divided. And then number two, let's get the rest of the cell divided. Once we have enough nucleuses, nuclei, to fill each of the new cells, then we can pinch off and separate. So we've got 1, 2. We've got either nuclear division, not either. We do. That's what's happening. Nuclear division first, and then cellular division, or cytoplasm. You know what? I'm going to change that. I know you just wrote it and now you can hate me. It's fine. Cytoplasm. And this is also going to be called cytokinesis. So you get to talk to your moms and dads tonight about cytokinesis and sound all like smarty pants. Yay. All right, so let's take a look at what the types of nuclear division are, because there are two ways we can sort out our cards. We've got 92 pieces of genetic information. They can either get split into two piles of 46, or there's some other trickery that can occur as well. So in nuclear division, there are two types that we're going to be talking about, mitosis and mitosis. That is because I'm dumb and I should have written meiosis. Okay. This part of the unit, we're looking at mitosis. That's the stuff that I want to make sure that you're good with. And so during mitosis, we start with a diploid cell, we end with a diploid cell. We start with a somatic, which means body cell. We end with a somatic body cell. We start with the exact same thing that we end with. The only difference is now there are two of them. They are identical to each other and identical to the parent. Identical. Oh yeah, no, that's AL. Oh good, it's now on YouTube for all posterity to know that I am a moron. Identical. I think that's right. In meiosis, this is only going to be done in sexually reproducing organisms, and the whole point there is to cut down the number of chromosomes that you have to make cells that have half as many chromosomes, that when they get together, you're back up to the normal number. We'll talk about that stuff later. Does anyone have any questions so far? Again, pop them in the chat if you do, because I want to make sure that I'm not like way too much. I'll keep going, but if questions come up, we're all good, okay? All right, so we're gonna start with mitosis. Whole idea here is now we've got 92 chunks of information that need to be carefully sorted. We do not wanna end up with one cell of 46 and one cell of 46, but they're not the same 46, you know what I mean? Like we don't wanna end up with 5 or 6 aces in one pile and 2 or 3 in the other. Whoops, I ended up with all the kings over here, but we're fine. No, you don't want to play with that kind of playing card, and your cells do not want to have that condition. That is not compatible with life. Everything goes downhill if you don't have all the right chromosomes present in the cell. So let's start at the very beginning. A very good place to start, by the way, or so I've been told from the sound of music. All right, so pro. Right off the bat, let's go ahead and review some Latin, Greek, I don't know, pro means early or before. So if you're thinking about history, the pro-bellum era means the time before the Civil War. It's the only thing I know about history, so like you can be impressed, but that's it. For the prokaryotes, remember that karyo means nucleus, so prokaryotes evolved before the nucleus. Prophase is the first earliest stage that we're going to be going through during mitosis. And at the beginning of prophase, what we see is that the DNA is still in chromatin form. But by the end, those sister chromatids have formed. They've condensed up to make the pairs of genetic information, okay? But a bunch of other things are going on as well. So during prophase, we have the centrioles. Those are the little dudes that look like stars on your screen, like the yellow ones. And so this pair of centrioles are starting to make proteins. All right, so these are going to be really integral to making the proteins that are going to grab hold of the sister chromatids and rip them apart. It's very, very sad, actually. Anyway, I'll get into that later. But so our centrioles need to be at opposite sides so that they can each exert a pull from other sides of the cell. Kind of like our origin is where the DNA and prokaryotes kind of got to the outside to be able to pull the chromosomes apart. That's going to be what the centrioles are doing. In addition, as I mentioned, in terms of the DNA, there's a few things that is a lot different. So let's talk about what is going on with the nucleus during prophase. Most notably is that we're going to be able to see visible chromosomes. We're going to be able to see that chromatin has condensed. Chromatin condensed, whoopsie daisy. Into sister proteins. chromatids. Okay, so if you can see chromosomes in a micrograph of a cell, if you can see that genetic information and it looks like there's like string in there instead of just a gray blob, then you know that at least some part of the M phase is going on, because starting in prophase, we are going to have visible chromatids. You're not going to see each one. It's not like you can look in and be like, there's one sister, there's the other. It doesn't work like that, but at least they're visible enough that they can be distinguished. In addition, we can see that the nuclear membrane is like, goodbye, this has been fun, but I'm going to dissolve now. And the reason that's important is because we need to have the spindle fibers, those protein fibers that are being made from the centrioles, they need to have access to the chromosomes. Otherwise, they can't pull them apart. So the nuclear membrane has got to go. You ever think about words, like as you're writing them? Disintegrate, disintegrate just means to unintegrate. So integrate, coming together. Anyway, good times, right? Moving on. So we've got the chromatin, it is condensing. We've got this nuclear membrane that is like, goodbye, it's been fun knowing you. So keep in mind that everything that we're talking about until telophase, we have we have all of that happening in the cell. It's hanging around the mitochondria and the Golgi apparatus. Like it's all there too. It is not in its own little compartmentalized nucleus anymore at this point. And then finally, the last thing that we're going to see is that little ball there in the corner. He's gone. The nucleolus also disintegrates. It will reintegrate later, but for right now, it is in pieces. If you could just kind of jot down, am I going way too fast, way too slow? Please let me know if you need me to repeat anything or anything, okay? All right, pro means beginning, the beginning of mitosis, the beginning of that nuclear division where we start with a diploid cell and we end up with 2 diploid cells, right? The beginning of that process is going to be prophase. And then from there, we're going to move on to metaphase. Again, there's kind of an early and a late stage of metaphase that you'll sometimes hear referred to as pro-metaphase and then actual metaphase, but I'm just going to kind of consider them together. And so what's going on here is that the centrioles are continuing to produce those protein fibers that I had just mentioned, and those are called spindle fibers. Okay, and those spindle fibers are going to grow from both ends of that cell, and they're going to connect not only with the genetic information, but also with each other. And so what we end up with is spindle fibers that kind of grow in and either attach to centromere. So remember, our sister chromatids are being held together at the centromere. Ooh, that was strange. Each of her. Okay. So not only are they connecting to these centromeres, but they're also connecting to each other and they're continuing to grow. I can't get my hands to grow, but you get the idea. And pushing against each other. And the ones that have attached to spindle fibers, you'll have a spindle fiber attached from both centrioles to the centromere of the parasister chromatids. So if I were to kind of zoom in, it would look something like, you know, here's one, here's the other, and they're attached here. I don't know why I didn't just draw them attached. Nope. And so we're going to have a spindle fiber that attaches that way and another one that attaches that way, okay? And so what you're gonna have is this pushing and pulling of the spindle fibers from opposite sides that eventually is going to push and pull just enough that those sister chromatids get lined up in the center. And that's how you know you are at the point of metaphase, is that at metaphase, they're in the middle. That's how you'll remember it, right? Metaphase, middle. So during this point, sister chromatids, there we go. Centrome, centrosome, no, centromere. You know, sometimes your brain's like, you should work, and then sometimes it doesn't. Good story. Okay, so those sister chromatids are getting pushed into the center onto this invisible line that is called the metaphase plate. Metaphase middle. And so from there, we've now paired everything up, lined it up. So all we need to do is tug and separate, and we ensure that each pair of sister chromatids, which are identical, get separated and go to each opposite end of the cell. The result is going to be that we ensure that the two new cells are identical to one another because those sister chromatids were identical, and then those sister chromatids were what were separated. And the separation comes during anaphase. So think anaphase apart. So what we're going to see is that the spindle fibers, which are proteins, are going to get hydrolyzed. They're going to get broken back down into their individual amino acid components so that they can get recycled for something different. And that's going to shorten them. So the spindle fibers I really like anthropomorphizing. I don't think I'm saying it right. Okay. But I picture them just being like, retreat. Anyway, moving into opposite directions. And with them, they're going to be pulling one of the pair of sister chromatids. They're just going to pull one side of it. At this point, we no longer call them chromatids. We're back to calling them chromosomes. Okay. And then finally, remember, anaphase, apart, A, great. Finally, we get into telophase, and during telophase, we are finally going to see that the nuclei have gotten to their finished state, right? They have what they need in them. So a new nuclear membrane is going to form around them. And they're also going to go ahead and start to decondense to relax a little bit. And form chromatin with an N at the end there by the end of telophase 4-2. T for telophase, T for 2 nuclei that have formed by the end of this process. Now, at the end of telophase, if all that were to happen was mitosis, you would end up with a cell that had 2 nuclei in it. But thankfully, we have another process going on. The cytoplasm is also dividing, and that's where we get into cytokinesis. So again, cytokinesis is when the cytoplasm splits apart, and it doesn't necessarily go right after mitosis. As you can see in our picture of telophase, it's already underway. So usually by mid to late anaphase, you have cytokinesis starting. Okay, so hopefully that makes some sense. So in animals, cytokinesis looks like this. As if it's bad enough that it looks like a butt, it's got a fun name too, just to really make biology teachers everywhere miserable. Yay. Nothing like teaching a bunch of like teenagers that something that looks like a butt is called a cleavage furrow. And when you think of a cleavage furrow, really, it's just kind of like a rubber band that gets, okay, it's like a rubber band that the cell makes and is inside of the cell, but that just kind of pinches around the outside and squeezes and squeezes and squeezes until you have 2 cells. And the organelles that are all throughout that cytoplasm get distributed out before it is completely divided, and the result is 2 new cells. Happy cells. Okay, if you're a plant cell, again, repeating this, you are not a plant. You are not made out of plant cells. Stop it. Anyway, moving on. Moving on to plant cells, because they have this cell wall kind of in the way, they can't just pinch apart. And so instead of a cleavage furrow, what they're going to end up with is something called a cell plate. And that cell plate is just a whole bunch of carbohydrates and proteins that are all being brought to the center line of the cell, right about where the metaphase plate was. And all of those different organic molecules are assembled into a like proto wall, like an early wall that eventually will form a completed cell wall that divides the 2 cells that were once one. And at the beginning of interphase, the cell is able to push out on that cell wall enough to kind of get some growth happening. Otherwise, you would have a plant cell that then became 2 plant cells in the same size, and then 4, and then so on, that stays the same size. That wouldn't work. So there's a little bit of stretch there, not a whole bunch. Okay, I'm going to go ahead and